बहुत बहुत ज़्यादा आपका पैसा बच गया, आपका फिजिकल आना जाना वो बच गया, तो बहुत ज़्यादा इससे क्या हो कि अब हमारा क्या हो रहा है अभी जो मुंह के है स्वयं के है वो हमको डेवलप करने के लिए कि आप 60% तो आप ऑफलाइन और 40% आपको ऑनलाइन करना है, अकॉर्डिंग टू न्यू एनईपी
न्यू एजुकेशन पॉलिसी, राइट।
So how to framework create the framework all the subject, so resubmitting all the subject na all the with the JNU BHU शायद एक अलीगढ़ में AMU वालों ने भी अभी उन्होंने कहा कि हम माइनॉरिटी वाले हम नहीं कर सकते तो था, उनका भी इलाहाबाद हाईकोर्ट में केस चला, अदरवाइज सभी सेंट्रल से आ गए एक ही वन अम्ब्रेला में सब लोग इंटरेस्ट, तो अच्छा भी है। हम तो कहते हैं एक जैसा सिलेबस हो, सबको एक जैसा पढ़े, थोड़ा सा उसकी लोकेलिटी के हिसाब से थोड़ा चेंज हो रहा है भाई,
ज्योग्राफिकल जो अपना है उस हिसाब से थोड़ा,
हाँ उस हिसाब से थोड़ी 20% to 30% कर सकता है। अदरवाइज ये जो बीएससी ऑनर्स वगैरह किया तो इससे कि बाहर फॉरेन में जाने का उनको MS करता तो सीधे, अदरवाइज 3 ईयर कोर्स 1 ईयर कोर्स फिर करता, फिर PG के लिए करता MS के लिए, ये पॉजिटिव वो है कि कम से कम आप, लेकिन बिग प्रॉब्लम इशू इज दैट इतना बजट कहाँ से आएगा?
जी वही होता है
जो रेगुलेशन है कि आपको 40 स्टूडेंट PG में उससे ज्यादा नहीं होना चाहिए,
नहीं होने चाहिए,
25 से, तो ये बड़ा स्पेस इतने बाय इतने का होना चाहिए, ये इंफ्रास्ट्रक्चर है नहीं और हम ये कर रहे हैं, ये बहुत मेजर टास्क है।
बिल्कुल सही बात है, वो तो है। न्यू एजुकेशन पॉलिसी में इन लोगों ने बहुत नई नई चीजें तो लाई है, अभी देखते हैं कैसे इम्प्लीमेंट होते हैं। अभी वी आर फेसिंग वेरी सून, वी आर फेसिंग ऑटोनॉमी, टुवर्ड्स ऑटोनॉमी, तो उसका भी चल रहा है। आई वुड लाइक टू इनवाइट यू ऑन आवर ऑटोनॉमी का BOS, we are in the process of
ऑटोनॉमी मिल जाए ना, हम अपना डिजाइन करें वो करें और पूरा थोड़ा उससे बहुत फायदा होता है और टोटली आप अपने हिसाब से अपने, मेरे को पता है कि मेरा स्टूडेंट किस कैलिबर का है, मैं क्या उसको करना चाहता हूँ, मेरा उनका वो ज्यादा इम्पोर्टेंट है ना,
जरूरी है।
भाई चाइना ने इन्होंने यही किया कि अपने हिसाब से, जापानीज ने अपने हिसाब से उनको किया, तो अब हमारा पूरा ओवरऑल कर दिया, पूरा सिस्टम जो था वो पुराना, अब अभी उसको समझना और उसको इम्प्लीमेंट करना वो बड़ा बिग टास्क हो रहा है। तो कैसे हो पाएगा
जी,
मतलब जो भी हम लोग एक ट्रांजिशन पीरियड से गुजर रहे हैं पूरा बड़े मुश्किल तो हम CBCS सिस्टम को चॉइस बेस्ड क्रेडिट समझ पाए थे,
सही बात है,
12 क्रेडिट क्या है, तो अब यहाँ 10 क्रेडिट कर दिया उन्होंने।
जी अभी तो वो एकेडमिक क्रेडिट बैंक भी आने वाला है ये बच्चों के लिए,
वो तो आ गया, उन्होंने पिछले बार मोदी साहब ने कर दिया था।
हाँ, यूपी में तो हो गया ना शायद,
हाँ 1 ईयर हो गया, अब उसको सब इसमें इम्प्लीमेंट होना है। जी आइडिया तो बहुत अच्छा है, बहुत बढ़िया है, लेकिन बस यह है कि इम्प्लीमेंट हो कैसे पाएगा वो बड़ा बिग इशू है।
वो तो पहले 2-3 साल तो यही चलेगा, ट्रायल एरर पल्लवी मॅडम थोडा फास्ट करा मुलांना, आपण 1-2 मिनिटात विल स्टार्ट द सेशन।
यस मॅम,
ठीक है, shall we start sir?
मॅम
हाँ सर लेफ्ट झाले बहुतेक, परत येते। किती मुलं आली आहेत बघा. 40 स्टुडंट्स मला पीजीचे दिसत आहेत.
हा मॅम टी1चा पेपर आहे.
तेच. आणि रेकॉर्ड ठेवा आहे कोण कोण प्रेझेंट होतं कसं ते.
हो प्रेझेंटिंग त्यांची बी आहे.
स्टार्ट करते हैं सर.
हाँ हाँ
यस
मेरा जरा नेटवर्क इशू हो रहा था थोड़ा सा मैं चेक
जी
कर रहा हूँ
जी.
नेटवर्क थोड़ा
एफटीएच से कनेक्ट करेगा
हाँ हाँ
तो चलना चाहिए एफटीएच जी नहीं चलना चाहिए
ठीक प्रॉब्लम नेटवर्क तो चला जा रहा है चेक करो ठीक है दोबारा कंप्यूटर मास्टर और Okay, ma'am. I'm audible.
येस सर येस सर. स्टार्ट करते हैं. माझा आवाज येतोय पल्लवी?
यस मॅम.
Okay. Good morning and warm welcome Dr. Rajendra Singh Partiyal ji, Department of Zoology, Chaurus Campus, HNB Gadwal University, Gadwal. My staff member Akash Bagade sir, Pallavi madam, Yash sir, Pratiksha ma'am, and my dear students for the guest lecture today under DBT Star College scheme. Just now I spoke with Dr. Principal Abhay Khandagare sir. He told me that he will join if possible. He is busy with some office work. May I now call upon Pallavi madam to introduce today's speaker Rajendra ji Partiyal.
Thank you, ma'am. Welcome. This gives me immense pleasure to introduce our today's speaker, Dr. Rajendra Singh Partiyal, Fellow of Royal Entomological Society, London, visiting Associate Professor and Institute Fellowship. His research interests are systematic cytogenesis, molecular phylogeny, ecology, evolution, pest, and biodiversity of family Drosophilidae. He has given 38 species new to science and 18 new records, first time from the India. A total of 56 new Drosophila species from Uttarakhand, West Bengal. He has 24 years of research experience and 19 years of teaching experience. He has 34 research articles in well-reputed journals and has reviewed many research papers in different journals. He is also a member at Royal Entomological Society, London, International Society of Zoological Science, China, Vigyan Bharati, Uttarakhand, There is a lot more that could be said about him, but without taking lot of your time, I would request our guest to proceed with his talk. Thank you,
Okay.
sir.
Thank you. Thank you. Okay, it's my pleasure. Share my ideas, my research work. So I just okay. Now it is visible.
Yes, sir.
Okay. Okay.
Yes,
Thank
sir,
you.
it is visible. Yes.
Okay. Okay. Thank you. So today I thank to uh, Principal Sir, Abhay Sir, and Dr. Rasmi Maurice Ma'am, and other uh, our colleagues. So it's my great pleasure to uh, invite me in your online virtual mode in this platform. So I just share my ideas, what I do, what I did in my past year in with the Drosophila melanogaster. So first of all, why use Drosophila melanogaster as a model organism and how to handle it? First, how to identify the Drosophila and how to rearing from field to lab. So, as you know, in Gregor John Mendel in 1888 used the Python sativum pea plant as a model. So, in the 1911, T.H. Morgan and his colleagues used the red eye color insects. So, as you know about seven contrasting character in pea plants, seven contrasting character in pea plants. But uh, in case of Drosophila, about more than 100 characters, more than. Can you imagine? It's very small, about 2 mm to 6 mm, 4 or 6 millimeter in length. But it's very small, very small. So uh, this is our first slide. Second slide. In 1911, T.H. Morgan and his students, Steve Bridges, Mueller, and student, came across red-eye insect, fruit fly. Generally, its name is first name is banana fly, fruit fly, vinegar fly, or nowadays is called as garbage fly. So, is attracted through the fermentation of the fruit. So since then, it has made its way in research lab, helping scientists to explore fundamental problem in biological sciences, or especially genetics. In genetics, it is used as a raw material after Mendel's work. So Drosophila are a very versatile, very tiny, or low maintenance, or non-harming model organism, and they can be easily used in all field of life sciences like genetics, biotechnology, cancer biology, genomics, reproductive biology, reproductive ecology, developmental biology, microchemical study, ecology, immunology, many more. So very important thing is that there is three things: limited budget, limited space, or limited time. So very small lifespan is about average about 16 to 23 days. Is a life cycle. A life cycle is four stages: egg, larva, first star larva, second star larva, third star larva, pupa, then adult. And the whole life cycle completed within a 16 to 23 days. So if the temperature and humidity is high, then is life cycle completed within 10 days or if the humidity temperature is low, and its life cycle is about 3 months. So it's a very totally manipulate or totally life cycle according to your protocol. So such a model organism, we must learn capturing, rearing, and culturing their progeny, along with basic identification and differentiation between male and females. So first of all, if you collect the insect red eye color, but so many red eye color, how to identify the Drosophila? So uh, like Mendel's this understanding the tree plants, है segregation, dominance, independent assortment law. So you know these seven contrasting characters: round, wrinkled, yellow, green, pot color, height, axle, short, long. So all the experiment in tree plants, but in case of all the genetic experiment in tree plants in 80s, but in 90s, totally in 1911 onwards, this tree plant replaced by this model organism Drosophila. Drosophila melanogaster is a common cosmopolitan. ये स्लाइड आपको दिख रही है, इट इज विजिबल. हेलो
सर, दिख रही है.
Now, like in first picture, the whole insect, especially Drosophila melanogaster, is very small, 2 mm to 3 mm. But according to species to species varies. Like in case this right side, Drosophila lacertosa, it's about 6 mm, 6 mm. Highest. Or this Drosophila melanogaster very small in size. Body divided into 3 parts: head, thorax, and abdomen. If the red eye color, here first head, the red eye color, one character, first character red eye color. In second picture is dull red eye color and it's yellowish, but generally it's red eye color. First character. Second character, the wing venation pattern. Wing venation pattern, first anterior crossline, posterior crossline, and one costal breast. So with one satellite, one spine. So this pattern in every insect having their own wing venation pattern. Wing venation pattern is particular identification pattern of each insect. So and abdomen. So this thorax, scutum, scutellum, this bear consists of three pair of legs, foreleg, mid-leg, and hand leg. And wing, forward wing is totally developed. The hand wing is not developed. It converted as a halter. This halter dumbbell-like structure, 2-point. If you look at 2-point, it's a halter is as a balancing organism, balancing tool at the time of flight. So, in case of this fore wing, fore leg, in male case, sex com is here. So this and other character are brownish in color. Whole body generally is brown in color, but sometimes is blackish, dark brown in color. And the arista is generally plumose type, feather-like, plumose type. But in some cases, this arista or dumbbell says a butterfly like this is Fortica azima, Fortica whatever. This fly is my lab described in 2020 as a Fortica whatever. So this is a lacertosa. So if you go to field or so many types of, some is totally fruit, attracted by the fruit, sometimes is flower visitor, or sometimes is some mushroom visitor, so fungivorous flower visitor, so or sometimes is wild rose. So if you go, you will go to fruit market or in juice center or in any fruit product. So this have a small brown color, small. So when we collected this fly, generally in the particular area, or is this decision according to the altitude level above the sea level. In 300-meter different species, in 600 different species, 1,000-meter different species. In my area is Uttarakhand, especially this hilly region. We collected from some species in the plain area, Dehradun or some Rishikesh, then some Biasi or some Srinagar, 500-meter, or some Agasmuni, and there is Chopta, about 200-meter meadows. So, on the basis of altitude pattern, species varies according to altitude, one thing, and on the basis of fruit preference. So, two indicators on the basis of what type of habitat. So, this Impatiens plant. So, this is the differently is black color. This is the Lodifossa genus. So, different genus in right now about 7,122 or minus plus 4, 5 total number of species or species belong to 76 or sometimes 77 genera described in all over the world. But in India, about 348, according to my database, 348 species right now and 27 genera. So it's a very good variety, or it's a hot spot as a, according to the Drosophila taxonomy. So if you go in the some juice center, some fruit industry area, how to collect through this net shipping. So trap method, net shipping, or direct to the aspirator. So first character is red eye color. How to identify? Generally, first is Drosophila melanogaster, some amniota, then on generally Drosophila busci. This is a tiger fruit fly. We call it commonly tiger strip. Is a abdomen pattern. So on the basis of one red eye color, on the almost or red eye color, first this Drosophila whole Drosophilids, phylum Arthropoda, class Insecta, order Diptera, family Drosophilidae. These Drosophilidae I already discussed 7,122 species. This is 67 genera. So these family Drosophilidae divided into two subfamily, Drosophilinae and Stagnini. On the basis of this proclinate bristle or inner inclination bristle, proclinate bristle, on the basis of the distance of this bristles. So distance we know. So if the distance between this proclinate and their posterior, then it's a stagnini. If this closest two bristles are anterior, first or second is the closest, then Drosophilinae. So on the basis of the closeness distance between these three, divide into Drosophilinae or Stagnini. So the subfamily Drosophilinae is very diverse and different across 48 genera. Largest is Drosophila, 1,000 413 species. And subfamily Stagnini is a smaller one and is distributed across 28 genera. About Leucophanga is the largest. So in these two subfamilies, two types of Drosophila. One is you can say domestic Drosophila, one is wild Drosophila. Generally, it's all wild. But if those easily cultured, rearing in your lab, we can say there is a domestic. Domestic means you collect it from a domestic area, like a fruit market, generally is easily cultured. But if you collect from the forest area, or from wild area, from mushroom, from fruit, from flower, or from other uh, some tree sap. So that species not cultured, not easily cultured in your laboratory standard food medium. So first character is red eye color. Second character is wing pattern. First is red eye color. Second, wing venation pattern. What type of pattern? ठीक है ना? This R1, R1, first radial one, radial 2 and 3. Then this first cross line, second cross line. Or this 1, 2, 3, 4, 5, 6, 7. The total number of this. This is the, and there are some humeral causal brake or subcausal brake. This causal brake with the two small spines. Spines here. Right, actually. So it's a identification character. One is red eye color, second wing venation pattern. A particular pattern of wing venation. Then presence of arista. What type of arista? If this pedicel, post pedicel, this terminal fork, this upper branches or lower branches or dorsal branches or lower branches. So it's the number is fixed. When you identify the any species that is ventral branches is generally low or the low number or upper branches, dorsal branches high number. So this is the actual photograph. So now how first if you identify the this red eye color, then wing venation pattern. Third is how to identify the male and the female. Generally, in all the insects, in general perception, in all the insects, male abdomen generally is blunt, is banded or blunt, not sharp. Or tip is generally blackish in case of male, or small in size compared to female. And female generally pointed, sharp. In every the general case in insects, female is tip is very sharp. In this case, this is called ovipositor. This last point, of larger in size in compared to male. So if you look my cursor, this sex forms 2 and 1. This sex form generally a male character. Sex form tip of the abdomen is blackish and blunt, not sharp abdominal tip. But in case of this female, generally sharp, pointed. Through this, if this ovipositor is heavy serrated, a saw-like structure that punctures the soft skin fruit, like in case of cherry, grape, or other some fruits, apple. So is cut through this ovipositor. So it's a tool and inject the some egg inside the fruit at the timing of flowering stage. The egg hatch in the second one in flower. Then after fruiting, fruit is very neat and clean, but inside there's some egg larvae. So through injected through this ovipositor. So in my total area, during my research area, I cover this purple gray area. Uttarakhand is two division, Kumaon region and Garhwal region, two region, Kumaon and Garhwal. And my work is during my research, total I cover this Kumaon region. But after my postdoc, I joined my this HND Global in 2002. From since that in 2002, I explored uh, this Rudapyak, Chamoli, or Tehri, or some part of Dehradun. Now, one my student is from his Hardwar. So we explored from the Fudil area and to Himalayan region, Western Himalayan region. So next point, method of collection. How to collect from the field to lab. So one thing is that one trap bite method. If small Bisli bottle, any mineral water bottle, just create a window, cut this from the middle side, and insert the banana, and hang in the some bushes or shady area, humid area. After the 1 day or 24 hours or sometimes 48 hours, what type of condition of the banana? If the banana is very ripening condition, about 7 or 8 hours, you collect from some fruit fly or net shipping. If the very specific designed a small netting, cotton, handmade net, and collect in wild areas. If you are focused on the diversity of the insect. If you're hearing of the insect, it's a best way to installation of the trap bed in some paddy field area or some orchard or some fruit belt areas. है But in case of the you targeted the taxonomic work, diversity or host plants, you go visit to a forest area. And the third way method is direct collection with the aspirator. If there's some bark of the tree and some Ganoderma mushroom. Myself, I collected this Ganoderma, some is mycoderma, collected directly from the mushroom. So you directly collected through this, it's really designed this aspirator. So another method, one of my Japanese collaborator, Professor Majanori John Toda, especially designed in 1977 or 80. This box, here a tray of the banana with some mash with some yeast and a paste form. Then this bottle is contained with the 70% alcohol. Then this from this side, some attacked some fruit fly and captured here. So this plastic box. This is a specially designed trap box by Professor Toda. It's my student. And then through the insect net, it's designed. So collect with the help of aspirator. Then this I already discussed. So this, suppose this is Drosophila lacertosa. Lacertosa also culturable fly insect, and this fly is 2020 uh, explored described as a Fortica Watebi. This etymology Watebi, etymology means origin of the name. Totally about rare night about eight to all over the world. Watebi is about eight, but its eighth one is Watebi. My another Japanese colleague, Professor Hidachi Watebi, uh, he helped me in some leucophanga species. So he worked on all the Asian Drosophilids, or especially in taxonomic work or some metaphase chromosome. So this is Mycodrosophila gradiosa from this Ganoderma species mushroom. So totally is mushroom beta fungivora. This is flower from the flower, or this is the correct from the fruit. So if you visit in your forest area, how to collect? If there's some water catchment area or some stagnant water or some low stream water, so log pasted with this yeast and banana. After few hours, about 6 or 5 hours, some fruit fly attacked by through this banana. Then you directly over this, so collected through the net shipping or through the aspirator. So one kind of method is directly, not hanging any bottle, just or this through the net shipping method, or to, or sometimes in a bark area, very small fly, so directly collect through this. So in case of mushrooms, this mushroom, some particular genus like a Drosophila. One is not right now not described in communicated species, not published here. But this Drosophila mendelens, I collect my team collected from Mandal Chopta near to Badrinath or Kedarnath area. This collected. So this feeding only the mushrooms, but another one from this wild rose, Lodifosus, but K1 is not described right now. But another is invasive plant, Drosophila cubensis. First time new recorded, but my Japanese colony and some Spanish Perlech or whatever described this in 1997. But my team, this fly Drosophila whatever cubensis collected in I think 2013 or 14. So first, how to identify the Drosophila. Second, how to identify the male and the female, diagnostic of male and female. Third point, if you clear this level, collection, identification, then after the rearing of the particular species, then after the get a pure line culture. What is a pure line culture? Means if you collect about 50 female, you have no idea about the which particular species, but just you collect a Drosophila female. All the male deposited in the 70% 100% alcohol, for your further study for technical work, but only female fruit fly collected in standard food medium. Standard food medium is maize powder, brown sugar, yeast, agar-agar, nepazine, antifungal agent. Then 500 ml of water, some and some piece of fruits, banana, mashed with this, and is boiled and cooked. Then is pour in the vials. Then after second day, all the female collected and transferred in the particular food medium. After the few days, it means 1 week or 14 days, 2 weeks, some cracking in the food. That means the culture is well developed, well established. And then you collect the male fly from the particular wine. After that, dissecting the male genital organ. As you know, the in Drosophila, it's a mating system, locking system. A particular species, male species, a particular female species, mating on a particular particular species, yeah, own species. If the sibling species is crossed, then next species is not a sterile species, not a fertile species. So mutation rate is very high in case of Drosophila. So you first rearing, then pure line culture. Suppose you first start the some 50 wiles of 50 female. Then first generation. Then again you separate, only pick the only female. Then again in separate wild. Then virgin male or female in single wild. Then is so this is the pure line. Line number 1, line number 2, line number 6, line number 50. So these are. Then after that, dissect the through this stereosome microscope in 10 action or through the mounted needle. Then study the genital organs, especially this male female genital organ, especially the male, not consider female but especially the male. So this male genital organ match with the available literature. All the literature available in JDD, Japan Drosophila database, or some Drosotax, or some available in your lab, my laboratory. So match with this genital organ. If this genital organ not match with any relevant, very close but not very similar, in that case, that may be a new species. Then we further investigate the head region or wings or genital organ. So then simultaneously you need a microscope, stereosome dissecting microscope. Then and this microscope connected with MIPS or Dino capture camera. Eyepiece in this camera is attached with this eyepiece and with your laptop. Then finally is this shape of picture and you describe, like in case of Lordifosa Karva, described in Fartian and Tora in described in 2017. So this whole journey of the Drosophila, Fortica Watabe, described on the visit. So this paramere, adiegus, or genital male genital organ, is it a periphallic organ or is this a phallic organ? Now terminology change day by day. Now a consortium by the all of the world difference, a common terminology nowadays used after 2021. So culturing in the this this vials, this vial or tube, then numbering which particular line, and all the male cultured in male preserved in the 70% alcohol or 100% alcohol. So if you this is first starting of this vial for medium, then if the first star larva, you need a study of polytene chromosome, salivary gland. So if this cracking and some larva wall of this vial, first star larva is very small, second larva is long but very thin, but third star larva is very long and some puffy or very active. That indication is third star larva. If you collect this third star larva, put in a slide and dissect in isotonic solution, pH, isotonic solution, then two needle pull anterior posterior part, then salivary gland or oesophageal alimentary canal is pull out outside from the whole body cover. Then two balloon shaped structure is found, that is called salivary gland, and two small wall-like structure is a brain ganglion. Brain ganglion for the metaphase chromosome, है ना? 4 pair of chromosome through 1 pair of submetacentric, 2 metacentric, 1 telochromosome. So total 4 pair of chromosome, 8 number from the brain ganglion. If the salivary gland remove the fat body from the salivary gland and treated with 45% glycolic acid, then crushed by put the cover sleeve and crushed by the arrowheaded needle, and some ring shaped or some thread-like structure from each cell found, that is polytene chromosome. That was what then. So a species on the basis of food preference. Some common species, specific beets like banana. It is most commonly used bait, banana. That's why it is called banana fly. It could be used to collect cosmopolitan fruit fly, Drosophila melanogaster, and other commonly found Drosophila species. Tomato, adding yeast. If add some yeast to it to perform and attract a bait of Drosophila suzuki or Drosophila busci. Two common species, soft skin tomato fruit. Another orange, it also attract a bait Drosophila suzuki or Drosophila busci. Grapes with add yeast act as a attract the bait, especially Drosophila busci and Drosophila immigrants. Also Drosophila immigrants are cosmopolitan species. Or these species you collect throughout the year. It's a tolerance power, very cold tolerance, like in case of in Nainital, about 4 degrees centigrade in your kitchen, you collect from the kitchen area, or in if it is the 30 or 35. So it's a very high elasticity in the tolerance of the humidity and temperature. Drosophila melanogaster, Drosophila immigrants. Choose the vinegar bait, Drosophila suzuki help the trapping more flies, keeping bait in the moist feet traps, improve the collected yield. So if you go to field areas, especially wild fruits, some poisonous fruits, it's called a snake fruit, some poisonous fruit. If this ripening is start from the tip, then it's soft skin. But is one species is Colchosomia. Colchosomia species collected from this field, from this fruit fly, wild flower, and the invasive flower, all generally in the Lodifosa, genus Lodifosa, or some Heterodrosophila, and from mushroom, some Ganoderma, Mycodrosophila species. Or this apple orchard, some Drosophila Suzuki. Nowadays, Drosophila Suzuki and the Japanese indigenous declared as a pest, generally destroy in Philippines, generally destroy the banana crop. In this cherry in USA, UK, destroy the cherry, and in our area, Japanese indigenous destroy the all apple, damage the physical damage, economical loss for the horticulture farmers. So finally, on the basis of the identification, how to if you PG level student, if you will opt to pursue your PhD work after few next year, so how to uh, summarize your work, how to publish your work. So just you review all the past literature, available literature in your website, or through the expert comment. Then you publish their work in a systematic way, in particular high resolution pictures, good quality content. Then publish this work this way. So this salivary gland, polytene chromosome, two type of giant chromosome, Lambrus chromosome or polytene chromosome. Polytene chromosome collect from the all the difference, or especially chiron. First time Balbini collect from the chironosomni. Salivary gland is outside of the larvae, but in Drosophila melanogaster, third star larvae inside there, only one pair, but in the chironosomni is two pairs. So total number of 6 arms. Lambrus chromosome oocytes from the amphibians, but polytene from the all the dipterans. This. So this one portion of my study, during my PhD work, this polytene chromosome. This is the nucleus organism region. So total number of 6 arms. So it's believed that in case of metaphase chromosome, this is a metaphase chromosome collected, prepared from the brain ganglion, cerebrum, brain ganglion, or this from the salivary gland. This eight chromosomes, 4 pair of chromosomes, these all the centromere join to go together and create this chromocenter and 6 arms, or each arm identify on the basis of tip of the arms. First, second left, second right, third left, third right, or X, a small arm. So it's believed that these bands, interbands of pop region or some inversions. So it's a how to chromosomal mapping. This technique. This is straight line. Each arm is separate and divided into particular measurement, and the total number of bands. Count the total number of bands, total number of interbands, total number of pops. On the basis of this, to identify the particular species. Nowadays, on the basis of mitochondrial DNA, molecular taxonomy, we morphological taxonomy correlate with the morphological taxonomy. They collect the isolate the DNA, or it is a mitochondrial DNA and CO1, CO2, NADH. Then these particular genome, a particular base pair in a particular species. Suppose in Drosophila melanogaster, for example, 613, 651 base pair. In Drosophila Suzuki, 650 or something like that. So particular is a particular base pair. So on the basis of in nowadays again cross check a particular species. Suppose you declare the some Drosophila syrianegrensis, a Drosophila punensis. Suppose if anyone will collected one Drosophila from Pune. So name, taxonomic name, ensis, locality, type locality, ensis, punensis, Drosophila punensis. So Drosophila punensis, so a particular length of the base pair of the mitochondrial DNA on the basis of linear DNA, nucleus DNA, and the mitochondrial DNA. Compare, then declare the new species on the basis of. So my collaborator, Professor, long collaborator in I think in 99 or 98, maternity tutor, and now that time is the Institute of Low Temperature Science of Japan. Now it is Emeritus Professor Hokkaido University Museum. Another my Chinese collaborator Jiang Jung Gao, Yuan University. Some my impatible, some flower visitor impatible flower insect collected from the my team from this Himalayan region, Badrinath Kedarnath belt, and same wise this my Chinese collaborator selected from the Tibet area, same species, impatible flower, flower plant, impatible genus. One another is my collaborator nowadays, Dr. Taru Kotaha, Division of Diversity. He is an expert of molecular taxonomy, molecular phylogeny. Earlier, he is as an assistant professor in Tokyo Metropolitan University, Japan. Another my collaborator, Dr. Subhas Raj Kohit. Now associate professor in Ahmedabad University, and one my another Kishore Kumar Gupta, HOD Department of Zoology, Binawa University, Hazaribagh, Jharkhand. So last, if anything is endangered in India, it's our taxonomist. In taxonomist, in the backbone of the diversity, whole diversity. If plant taxonomist, animal taxonomist, insect taxonomist, so a founder member of Zoo Outreach Organization, Sally Walker, quoted this: if anything is endangered in India, it's our taxonomist, but not in India. I include all over the world. All the dipterologists, taxonomists, hepterologists, all the taxonomists work almost finished. But we need some enthusiastic or energetic worker, especially explore our diversity on the basis of a taxonomic point of view. My Japanese collaborator emeritus Professor Choda, Professor Brain Kumar Singh, my mentor. Now he is retired. Myself, my first student, Dr. Manisha Sarswat, Dr. Pradeep Sati, Mohammed Jain Khan, now is assistant professor in Government Degree College, Asa, three, Sonali, Khali, Pragya Topal, and Sanita. Now is registered. Four is awarded and three is registered. Ma'am,
Yes, sir. Yes, sir.
okay. Any queries or any?
Yes. Any question from student side?
If any, किसी को कुछ पूछना है तो बोले. अगर आप लोगों को थोड़ा सा कर सकते हैं, मैं आपको अगर बताऊं, थोड़ा सा बिस्लरी का बोतल होगा ना, आप इसको ऐसा करके, ठीक है, और एक विंडो बना लीजिए. जैसे कट करके, ये कट किया, इसको विंडो बना लिया. स्पेशली मैं पीजी लेवल एमएससी स्टूडेंट है
जी सर.
साइटोजेनेटिकल वर्क है ना, पॉलिटीन क्रोमोसोम बनाने का आप बहुत इजीली, मैं आपको वीडियो वगैरह भेज दूंगा अगर बना आप लोग इंटरेस्टेड हैं तो. इससे आप एक विंडो बना दीजिए इसमें, और इसके अंदर बनाना फिट कर दीजिए. इसके अंदर पूरा बनाना चला जाएगा, और इसको फिर इसको हैंग कर दीजिए. इसको अब कवर निकाल के इसको हैंग कर दीजिए. हैंग करने के बाद आफ्टर 4 or 5 आवर आपका इसको हैंग करेंगे तो इसमें जितने भी हैं आप जस्ट उसको निकालिए और यहाँ पे टेप लगा दीजिए. और फिर लैब में आके यहाँ से आपको निकाला और यहाँ एस्पिरेटर डाला और यहाँ वाइल डाली, दूसरा वाइल ले लिया, दूसरा बोतल ले लीजिए और उसमें रख, तो आराम से इसको आप अपने घर में किचन में कल्चर कर सकते हैं फ्री ऑफ कॉस्ट. आपको कुछ नहीं कर, एक बनाना को आपको घर में मिल जाएगा, बोतल मिल जाएगी, और इसी को ऐसे रख दीजिए. उसमें जैसे कल्चर आ जाते हैं, फ्लाई आ जाती है कई सारी, तो उन फ्लाई को कलेक्ट करके आपको क्या करना है, जस्ट कलेक्ट करके उसको आइडेंटिफाई करना है. जनरली उसमें फ्रूट फ्लाई आएंगी. अगर आपने बनाना रखा है तो 100% उसमें बनाना रखा है तो फ्रूट फ्लाई आएंगी. तो उसका रेड आई कलर, उसको माइक्रोस्कोप में लाइए, उसको जस्ट आप स्टार्टिंग बिगिनर है तो लैब में रखिए, अल्कोहल में 70% या 100% अल्कोहल में रखिए, उसको देखिए. एक बार देखिए, 2 बार देखिए, फिर उसमें मेल फीमेल के सॉर्ट आउट करने की देखिए. और कभी मेरे को मौका मिलेगा मैं फिजिकली आके आपके यहाँ पे जो ड्रोसोफिला लैब एक सेटअप करने की कोशिश करूंगा, और आपको फील्ड वगैरह विजिट वगैरह आके इस टाइप का अगर भी होगा, अब हम नॉर्मल हो जाएंगे थोड़ा दिन में, तो कोशिश करेंगे अगर ऐसा हो पाता तो.
जी जी.
ठीक है. अगर कोई किसी का अगर पेरेंट्स है या कोई भी है बद्रीनाथ केदारनाथ घूमने आते हैं तो HND यूनिवर्सिटी आपको वे में पड़ता है. मेरे यहाँ से हम लोग जहाँ पे हैं भी HMB गढ़वाल सेंट्रल यूनिवर्सिटी है, 100 किलोमीटर केदारनाथ, और अब वो 200 और 175 किलोमीटर बद्रीनाथ, तो वो रास्ता है. और आप मेरे लैब को ज्वाइन कर सकते हैं 1 वीक के लिए, 1 मंथ के लिए, या 1 or 2 डेज के लिए. तो मैं आपको इस टाइप का आपका करके आप अपने लैब में जाके इसको इजी कर सकते हैं. और पॉलिटीन क्रोमोसोम, मेटाफेज क्रोमोसोम तो बहुत इजीली आप अपने लैब में बैठ के कर सकते हैं. तो मेरा हम्बल रिक्वेस्ट होगा, आप आज ये लेक्चर के बाद जरूर एक बनाना अपने किचन में रख दीजिए. और आजकल तो जैसे आपका मार्च वगैरह हो रहा है, और इसका है फ्रॉम मार्च फर्स्ट वीक, मार्च फरवरी लास्ट आपका बॉम्बे में, महाराष्ट्र वगैरह में तो आपका थ्रू द ईयर मिल जाएगी. देखिए हमारे जो जहाँ हिमालयन रीजन है, जहाँ स्नोफॉल होता है, यहाँ पर हम फिर अक्टूबर से हमको मिलना शुरू बंद हो जाती है, तो डायपोज स्टेज में चला जाता है इंसेक्ट पूरा. अक्टूबर, नवंबर, दिसंबर, जनवरी, फरवरी से मिलना शुरू हो जाता है. तो ये हर मंथ में अलग अलग स्पीसीज मिलेंगी और एक पर्टिकुलर स्पीसीज मिलेंगी. और अगर जो कोई अगर मेरी हेल्प कर सकता है, एक कोलकोसो में जो स्पीसीज में बात कर रहा हूँ, वो एरिएसी फैमिली में, अगर जो कुछ लोग जिनकी बॉटनी रही है, एरिएसी फैमिली एक फ्लावर कीप जैसा व्हाइट कलर का फ्लावर होगा, उसमें हैच करके चले जाते हैं. तो उसको कलेक्ट करके अगर मेरे को भेज सकते हैं तो मैं आइडेंटिफाई करके आपको उसमें आपका कलेक्शन और आपका नेम पे उसमें हम वो कर सकते हैं, ठीक है ना। किसी को
ग्रेट
कुछ
ग्रेट
पूछना
सर,
है।
बहुत अच्छी
हाँ
अपॉर्चुनिटी दी है आपने बच्चों को।
हाँ
एनी क्वेश्चन फ्रॉम स्टूडेंट साइड? कोई बात नहीं. Anyways, it was a wonderful experience, sirs, to see the rich diversity of Drosophila from Uttarakhand,
या
and very nice to know about your work on this tiny angel Drosophila. I'm saying so because what we can see in the field of genetics hereditary, it is only because of this species,
या
and very happy you are. Anytime you can visit his lab, you can see, you can. Have you can discuss if you are interested in doing research in genetics. So this is what I think the aim of such kind of the lectures that we organize for our students.
या
Over to you, Pallavi madam. I'll talk to sir later on. सर से मेरी तो फोन पे बात होती रहती है, बहुत अच्छा लगा सर.
नहीं, अगर किसी को भी मेरी लैब ज्वाइन करनी है, आप ऑफिशियली आपने मैडम से लिखवा के लिया है कि हमको यहाँ जाना है, तो हमारा
जी जी,
गेस्ट वगैरह है तो अवेलेबल हो जाएगा, कोई एकोमोडेशन की कोई इशू नहीं है।
ग्रेट
जब भी
ग्रेट.
बस केवल जो आपका यात्रा सीजन होता है उसमें बहुत पैक रहता है,
जी जी,
अदरवाइज जो ऑफ वो है उसमें आप आ सकते हैं, कोई ऐसा इशू नहीं है, मिल जाएगा।
ग्रेट ग्रेट. और क्या
नहीं
चाहिए
तो
हम बोलेंगे, और क्या चाहिए?
नहीं तो मेरा घर तो है अवेलेबल है।
जी जी. बस ये बच्चों ने काम करना है, सीखना है कुछ नया,
हाँ
एक्सप्लोर, जितना खुद को एक्सप्लोर करेंगे उतना ज्यादा आगे जाएंगे, मेरा ये मानना है बच्चों.
जाएंगे। हमारे
जी
जितने भी बच्चे हैं हमारे यहाँ पे, जितने ज्यादा पोस्ट जॉब करके बाहर जा रहे हैं वो लोग,
जी जी
और
जी
इसलिए अगर आप लोग नया फील्ड को एक लेते हैं, नो और कोई जिस फील्ड को कोई टच नहीं करा उसको करें, तो उसमें अपॉर्चुनिटी मिलने की पॉसिबिलिटी
जी
90% हो जाती है ना।
जी जी
और जो जिसमें सब लोग काम कर रहे हैं उसमें आप भी काम कर रहे हैं, वो थोड़ा वो आप एक अलग हट के अपना रास्ता अलग
रूटीन.
बनाए, तो वो आपको 100% एक
जी
आपकी
जी
आइडेंटिटी होती है। आज मेरे को नॉर्थ इंडिया में कम से ड्रोसोफिला टैक्सोनॉमी के तो मुझे जानते हैं, तो मेरे को
ग्रेट
अच्छा
ग्रेट
लगता है, भले
ग्रेट.
कोई काम नहीं कर रहा है।
जी जी जी, यस यस, बहुत बढ़िया बात है सर. और मुझे
हाँ
लगता है इस तरह का जो अगर नेटवर्क हम लोग के बीच में होता है, इससे बच्चों को फायदा ही होता है. देखिए
हाँ, फायदा होगा, फायदा होगा।
जी जी. और मैं तो चाहूंगी ये बच्चे जो है हमारे समर में इनको वेकेशन होते हैं, विंटर तो पॉसिबल नहीं है आपके यहाँ पे, तो
हाँ
उस
हाँ,
टाइम ये जब वेकेशन होता है उस टाइम ये लोग ज्वाइन कर सकते हैं या?
ज्वाइन कर सकते हैं।
जी जी जी.
हाँ।
ओवर टू यू पल्लवी जी.
एक्सचेंज प्रोग्राम है, आप जा सकते हैं, आप आ सकते हैं, ऐसा कुछ
हाँ,
नहीं
इस
ना।
तरह की जो गाइडेंस
हाँ
होती है बच्चों को, जैसे
हाँ
आप रॉयल सोसाइटी ऑफ लंदन के
हाँ,
फेलो है, या
हमारे यहाँ
ये
रॉयल सोसाइटी में,
एक्सपीरियंस?
हाँ मैम आपने अच्छा को याद दिलाया, रॉयल सोसाइटी में अगर जो बच्चे इंटरेस्टेड हैं रॉयल एनिमल सोसाइटी को, उसकी आपकी मेंबरशिप होती है। अगर
जी.
आप मेंबरशिप नहीं भी हैं तो उसमें इंसेक्ट वीक एक होता है और एक कंपटीशन होता है, फोटोग्राफ कंपटीशन।
ग्रेट
उसमें
ग्रेट.
इधर आपका अगस्त में होता है, ऑल ओवर द वर्ल्ड में। आप जैसे अपना मोबाइल पकड़िए, कोई भी इंसेक्ट है उसको क्लिक करिए, उसका नेचुरल होना चाहिए, जो उसका नेचुरल जहाँ उसका हैबिटेट है वहां पर आपने कोई इंसेक्ट खींचा, आप उसका किस क्लास का है, कौन सा इंसेक्ट है वो भी नहीं आता, आप डजंट मैटर, बस जस्ट आप उसकी 3 पिक्चर आपको अपलोड करनी है जेपीजी फॉर्मेट में। कहाँ से कलेक्ट किया है, कौन सी एरिया था वो कलेक्ट करना, और अगर उसका पिक्चर बहुत बढ़िया है, उसका जो वो एंगल वगैरह ठीक ठाक है, तो
जी.
कंपटीशन उसमें आपको 100 यूरो फर्स्ट प्राइज और 50 यूरो और 25 यूरो ऐसा कुछ मिलता है। तो 25 यूरो का कम नहीं हो तो बहुत अच्छा होता है।
जी जी
ठीक है
जी
ना?
जी.
तो ऐसे उसमें हर साल होता है, वो आप रॉयल एनिमल सोसाइटी की विजिट करके आप लोगों को कंपटीशन में पार्टिसिपेट कर, कोई भी कर सकता है,
जी
अबव 18 और बिलो 18 2 कैटेगरी है उसमें।
जी 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 जी. नाइस. ओवर टू यू पल्लवी.
Yes, ma'am. Am I audible?
यस.
Thank, thank you, ma'am. On behalf of PDS, Professor Ramkrishna More, College Akurdi, DBT Stars Team College, I would like to thank our today's guest, Dr. Rajendra Singh sir, for giving on lecture why use Drosophila melanogaster as a model organism and how to handle it, and explaining the different species of Drosophila, their culture and handling methods in very easy and simplest way. Uh, I would like to say thank Professor Rashmi More, ma'am, overall coordinator, DBT Star College Scheme, for organizing such a wonderful lecture. I also uh, I like to say thank you, Mr. Honorable Mr. Dr. Abhay Khandagre sir. They always motivate us to arrange such a type of interesting lectures for students of basic sciences. I also like to say thank of all my colleagues and other staff members, and the last but not least, thank you to all dear students to participate in this wonderful lecture. Thank you to all.
Thank you, ma'am.
Thank you, sir. Thank you so much.
Thank you. Thank you.
Yes,
Namaskar.
yes.
Okay.
Thank you. Okay.
Thank you.
फिजिओलॉजी सगळं आहे त्याच्यात बघ याच्यामध्ये तर पण दिसत कुठे ना कुठे असेल भाऊ पुढे बघा त्याच्यातच असतं डन 2 मिनिटाचं काम असतं लोड नाही घ्यायचं टाकले कुठलं वाक्य कुठं स्टार्ट होते तेच काही झालं सिम्पलेस्ट साधं सिम्पल होतं मस्त मध्ये लिहिलं की बाबा फॉर हिज व्हॅल्युएबल टाइम टू आवर स्टुडंट्स झालं फॉर गिव्हिंग a lecture on why Drosophila melanogaster and explain this to the person in very easy and simplest way with such efforts एवढाच वर टाकायचा होता इथं फक्त to our students with such efforts झालं बाकीचं तर सेमच होतं तुझं जे आहे ते तू दिस म्हणले सच तेवढं वर्ड ऑनलाईन शरस होते तिथे 2 हे आहेत ना